ago, that David set the stone for the relationship between transpiration and plant production and demonstrated that yield increases with the amount of water used through transpiration. But water is becoming sparse worldwide, therefore there is a strong need to improve the efficiency of water use. Based on earlier research at Virginia Tech and more recent work by my graduate student Sayantam Sarkar, I will present some aspects of peanut transpiration in rain-fed production in a sub-humid environment in southeastern Virginia. This is my talk outline. I will touch upon the problem, hypothesis, methods, observations, interpretation, and finally conclusions. It was shown that to grow well and produce adequate yield, peanut needs 1 to 2 inches of water distributed uniformly weekly from planting to digging. If not, then probably the fields will just look like in these images with severe wilting and pegs that cannot penetrate the soil. One would think that Virginia has plentiful precipitation, and indeed Virginia, North Carolina, and South Carolina are considered sub-humid environments. But based on the multi-annual average rainfall from 2000 to 2017, it is not quite the case. Rainfall may be sufficient early on in vegetation when only 1 inch of water is required every week, but not later on at beginning seed maturity. And therefore, it is no surprise that the response of yield to precipitation and irrigation is just like you can see in this image. Others have shown that some genotypes can control, limit their transpiration when vapor pressure deficit exceeds 2.5 kilopascals. And by doing that, soil moisture can be conserved early on in the season for later parts of the season when precipitation is not abundant. However, there could be negative effects as well. For example, reduction of transpiration, which evidently is achieved through partial stomatal closure, can limit CO2 availability for photosynthesis. But probably the most essential concern here is that these type of experiments, they were done primarily in growth chambers and not in the field. So the objective for this demonstration is to assess the transpiration rate and the relationship between transpiration rate and vapor pressure deficit in field as we grow peanuts in a sub-humid environment in Virginia. For this, I have used different sets of experiments. For example, I'm going to show data from research done in peanut variety and quality evaluation replicated test in 2009 and 2010. Here I'm going to compare two sets of genotypes. I will show data from 3 genotypes for which we monitored canopy temperature depression, which is the difference between air and canopy temperature, in replicated plots. CTD is an estimate of stomatal conductance there for transpiration, but doing so and using non-contact thermometers allowed me to monitor transpiration 24 hours a day for about 10 days at the full seed growth stage. And then for all the genotypes in the PVQE test, 30 of them, I will show data of transpiration measured using a photosynthesis meter during 11 and 2 p.m. in the afternoon. For this particular set of data, vapor pressure deficit was derived from air and temperature and relative humidity measured next to the plots. I will also show you data from another test conducted by a graduate student in 2013 and 2014 under rainout shelters. In this particular case, irrigation was used on 6 genotypes where transpiration, which were used to measure transpiration at 4-time intervals where the probability of vapor pressure deficit to change was high, from 10 to 12, from 12 to 2 p.m., 2 p.m. to 4, and 4 to 6. We also used a photosynthesis meter. Measurements for from 2 plants per plot were taken every other day from mid-July to mid-August. Again, plants were maintained under optimum soil moisture by weekly irrigation, and vapor pressure deficit was derived from air temperature and relative humidity recorded next to the plots. Finally, I will show data from a rain-out shelter test conducted by graduate student Sayantan Sarkar in 2019. Here we are going to look at the response of transpiration to VPD under 2 water regimes. He looked at 28 genotypes that were measured at 2-time intervals where the VPD was expected to change from 10 to noon and noon to 2 p.m. In the afternoon. He used the Lycor photosynthesis meter. Measurements were taken from 2 plants per plot from each water regime and from each of the 3 replications. Moisture in the irrigated part of the test was maintained by rainfall, but also by irrigation as needed. Vapor pressure deficit was also derived from air temperature and relative humidity recorded next to the plots. Okay. Based on the measurements in 2009-2010 on 3 genotypes and based on the diurnal transpiration estimated from canopy temperature depression, we can see that as the temperature and VPD increases, transpiration rate estimated from CTD also increased, and that was primarily from 8 to 10 in the morning when VPD went from basically 0 to 1.5 kilopascals. Then CTD decreased at the midday, even though VPD went from 1.5 to about 2.5 kilopascals, and then increased again in the afternoon until about 3 p.m. when it started to decline 1 hour after VPD began to decline as well. For all 30 genotypes measured in PVQE trials, during the elevated VPD time, which was between 11 and 2 p.m., the transpiration of all 30 genotypes decreased regardless whether the conditions were after a drought stress, before the drought stress, or during the actual drought stress. Each data point is an average of 3 replicates. Pre-stress, for example, represents a period with 10 days of no significant rain event at the beginning PEG stage. Stress period was after 20 consecutive days with no rain, and peanut was at the beginning pod stage, and pod stress was after the plots received 35 millimeters of combined rainfall and irrigation in 2009. Results from 2013-2014 trials showed also that all 6 genotypes had a negative response of transpiration to increasing vapor pressure deficit. So each data point here represents all time intervals that were measured starting from 10 a.m. to 6 p.m. through the day, and evidently the sign shows that stomatal conductance decreased when VPD increased, therefore transpiration decreased as well. Now, the degree of decline was more evident in the cool year 2014 than in the dry and hot year 2013, and this can be seen based on the value of the slope of regression of transpiration in response to VPD, which was almost double in 2014 versus 2013. And finally, the data from 2019 in irrigated trials and in drought trials measured on 28 peanut genotypes. So here we used VPD calculated, the blue dots calculated from the equipment itself, and actually this calculation is based on the leaf temperature that because of boundary layer conductance could be different, and also VPD levels could be different than that of the air, and also the red dots represent VPD calculated from the weather station. In irrigated plots, indeed, variation of VPD was from 1 to a little over 1.5 kilopascals, and we noticed a positive increase of transpiration in relation with the VPD, this in irrigated plots. However, when VPD was based on the actual leaf temperature, first of all, the value was much larger, anywhere from 1.5 to 3.5 kilopascals, so a lot more deficit at the leaf level than in the air, and the trend was negative. The response of the plants to increasing VPD was by reducing transpiration. In drought stress plots, measurements were done at the same time, one day on irrigated plots and one day on the drought stress plots for 4 weeks. Regardless how VPD was measured, the response of transpiration to increasing VPD was the same, and the response was linear and negative. So therefore, based on the measurements across years, different sets of varieties, using different ways of measuring transpiration and different ways of calculating vapor pressure deficit, we noticed a negative linear relationship between transpiration rate and VPD, in particular during the hottest part of the day from 11 to 2 p.m., when VPD is usually highest. These results agrees with the classical model of partial stomatal closure in response to midday temperature and VPD increase, and it was regardless of the temperature, soil moisture content, and apparently all peanut genotypes expressed this midday limited transpiration when grown in the field. Therefore, expression of limited transpiration did not explain yield variation among genotypes in rain-fed production in a sub-humid environment. Therefore, my question is whether the beneficial to drought tolerance coming from limited transpiration in peanut production exist.